Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CUET entrance exam question paper for the subject zoology conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of various subjects, you can go to my channel Omni Bioentrance. In the channel playlist C for CUET, and there you will find. So coming to the first question, it is match the following hormone with function. Leptin. Leptin, it suppresses appetite. So A2 is there only in option 4. So that must be your answer. So A2. Then pancreatic polypeptide, it inhibits somatostatin secretion. That is also correct. Okay. Then parathyroid hormone promotes formation of calcitrol. That is also correct. Epinephrine in skeletal muscles is conversion of glucose to glucose 1-phosphate. So it is A2, B3, C4 and D1. Answer is option 4. Then place these steps in temporal order, first to last. So here they have given the neurotransmission mechanism. So here this is your presynaptic neuron, this is your postsynaptic cell or the neuron. These are voltage gated CS2 plus channels. These are the synaptic vesicles. This is the one which is synaptic vesicle fusing with the membrane and releasing acetylcholine receptors. These are accepted by the receptor of the postsynaptic cell. Then there will be opening of the Na plus channel and influx of sodium ions, and resulting in the generation of postsynaptic potential. Okay. So the first step will be opening of voltage gated Ca2 plus channels in the presynaptic neuron. So C first is there in option 1 and 4. So 2 and 3 you need not see. Okay. After opening of the voltage gated C plus channel, you will have to go for exocytosis of synaptic vesicle. So these are the synaptic vesicles. So these are the synaptic vesicle. They will fuse with your plasma membrane and they will result in the release of acetylcholine receptor into the synaptic cleft. Okay. Then comes B, binding of neurotransmitter molecules to its receptor. So receptor is present on the postsynaptic cell, and these receptors, they will bind the neurotransmitter. Then what will happen after binding? Voltage gated Na plus channels and open, and influx of sodium ions will result, that is D, and then there will be generation of the postsynaptic potential. So the answer here is C, A, B, D and E. So 1 is your answer. Then which of the following is not true with respect to control of respiration? Okay. So peripheral chemoreceptors respond to changes in oxygen and carbon dioxide concentration in the blood. Yes, that is correct. They respond to both oxygen and carbon dioxide concentration, and thereby it will regulate the breathing to maintain homeostasis. So that is correct. Respiration responds to smaller changes in the blood concentration of oxygen than carbon dioxide. That is not correct. It is to small changes in carbon dioxide than to oxygen. That is, respiration responds more readily to changes in carbon dioxide concentration than to oxygen concentration. Okay. That is, even if there is a small change in the carbon dioxide concentration, it is going to affect the pH of the blood. And the various chemoreceptors in the brain, they will detect these small changes in the carbon dioxide concentration, and they will trigger responses to maintain acid-base balance. That is what is happening. So as per me, they have asked the following is not true. So that is your option 2 is the answer as per me. Then chemoreceptors in the CNS respond to changes in carbon dioxide concentration in the blood. That is correct. I told you now. Okay. So that is correct. Central chemoreceptors are sensitive to changes in pH of the cerebrospinal fluid. That is also correct. So as per me, answer 2 is the answer, but as per your answer key, they have given 3 as the answer, which might be a printing mistake. Okay. Then next one. The Asian mongoose and American skunk evolved independently of each other. So that is the key word there. But show similar structures and features. That is another keyword. Example, ability to spray their attacker with musk. These similarities between the two organisms are most likely the result of. So here it is the case of convergent evolution. Because convergent evolution, the organism evolved independently of each other, but they show similar structures and features. So the Asian mongoose and African skunk, they evolved separately, and even though their evolution was separate, they showed similar structures and features, that is ability to spray their attacker with the mouse. So it is the case of convergent evolution. Answer is option 4. Then a genetic drift, it is random changes in the gene frequency over time. Divergent evolution, here 2 or more related species, they evolved different traits or different structures or features. That is divergent evolution, which is opposite of what is given here. So it cannot be the answer. Allopatric speciation is when two populations become geographically isolated. So the answer here is convergent evolution. Then rifampicin, an inhibitor of RNA polymerase, is added to bacterial culture. The culture is immediately divided into 3 fractions to which radioactive thymine or uracil or methionine are added. Okay. 5 minutes later, the cells are lysed and the radioactive label in all macromolecules is monitored. Now they have given, what will you observe? That is the answer question. They have given rifampicin is an inhibitor of RNA polymerase, and you know RNA polymerase, it is involved in the synthesis of RNA from DNA, that is in transcription. So once rifampicin is added, RNA polymerase activity will be inhibited. So RNA synthesis will be inhibited. And RNA, you know, it is having uracil, not thymine, as in case of DNA. Okay. So it is uracil. So uracil will be incorporated less because RNA polymerase is inhibited. Then thymine and methionine, there won't be much problem there, because thymine means it is involved in DNA replication, it is seen in DNA. So what they mean by telling thymine is radioactive means DNA replication is not hindered, only RNA polymerase is inhibited, DNA polymerase is not inhibited. So thymine and methionine, methionine is for protein synthesis, it is an amino acid, translation is not inhibited by RNA polymerase. So thymine and methionine incorporate, radioactive label will be incorporated more in thymine and methionine than in uracil. So the first option, that is 1, is your answer. Then N-linked glycosylation of proteins in eukaryotes is initiated in the lumen of endoplasmic reticulum. If it is all linked glycosylation, then it would have been Golgi apparatus. Then next one. Arrange the following in order of their action. G1 cyclin CDK complex. So G1 cyclin, it is active during the G1 phase, and it promotes progression through G1 and prepares the cell for entry into S phase. So that will be the first one. A will be first. They have asked for the correct sequence. So A coming first is there in 2 and 4. So A. Then comes C, S phase cyclin CDK complex. So after G1, it is your S phase. So A, C. Now even if you don't know anything about APC CDH1 and APC CDC20, if you know A and C are the correct sequence, that is after A, C is coming, because after G1 it is S phase. If you know that, even then you can make your answer correct if you see option 2, because only in option 2 A and C are together, whereas A and D are together in option 4, which is not correct. Anyway, I will explain what is this APC CDH1 and APC CDC2. APC is anaphase promoting complex. So APC CDH1 and APC CDC20 have got something to do with the anaphase stage. So APC CDC20, that is the first one to come after S phase cycling in this question. And APC CDC20, it is activated during the metaphase anaphase transition, and it helps in the degradation of proteins such as securins and cyclins. Now what is this securin? Securin, it prevents sister chromatid separation until the onset of anaphase. That is what securin does. It prevents sister chromatid separation until the onset of anaphase. Until your metaphase, sister chromatids are joined together. They are not separated. Only after anaphase and spindle fiber formation do they separate and go to the separate poles. That is what is happening in your mitosis. So securin, it will hold the sister chromatids and prevent their separation until the onset of anaphase. So APC CDC20 is the next sequence, next one to come in the sequence. Then next one is APC CDH1. So this is activated during the late mitosis. So here the chromatids have got moved to the poles. So what this one does is this APC CDH1 does is it will help in the degradation of cyclins and other mitotic proteins, and thereby allowing exit from mitosis. Okay, that is APC CDH1 function. So answer here is ACDB. Then identify the correct statements related to sex-linked inheritance in humans. Males are considered to be hemizygous for genes present on X chromosome. Yes, males have only one X chromosome and the other is Y chromosome. So they are hemizygous. Then majority of sex-linked traits are carried on the X chromosome. That is also correct. Like color blindness, hemophilia, they are carried on the X chromosome. Genes that are not sex-linked are present in somatic cells and absent in germ cells. No, genes that are not sex-linked are present in both somatic cells and germ cells. 1:1 ratio of males and females in humans is the result of meiosis in males. As per me, that is also correct, because it is during the spermatogenesis the sperms get equal number of X and Y chromosomes. That is, X and Y chromosome bearing sperms are produced. And upon a fertilization with the egg, which is having only one X chromosome, either the sperm carrying X chromosome will fuse, or the sperm carrying the Y chromosome will fuse. So it is the meiosis in males that determines the ratio of males and females in humans is also correct, but that is not given in the option. They have given A and B only. So I am forced to go with option 3. Then the single X chromosome present in the father is inherited by the son. I told you previously also. The single X chromosome present in the father is not inherited by the son. It is inherited from the father. Only Y chromosome is passed on to the son. Okay. Then next one. Match the following. Diaphysis. Diaphysis is the long cylindrical main portion of the bone. A4. A4 is there in 2 options. Okay, 3 and 4. Now see B. Epiphysis. Epiphysis is the proximal and distal end of the bone. So B3, that makes option 3 as the answer. Metaphysis. Metaphysis is the region between diaphysis and epiphysis, and endosteum is a thin membrane that lines the internal bone surface. So A4, B3, C1 and D2 is the answer. Then again, species and identifying characters of the species. Allopatric species, geographical isolation. So A2, species inhabiting different geographical areas. A2 is there in 1 and 4. Okay, we will see. Sympatric species, species inhabiting same geographical area. B1 is there only in 1. So option 1 will be your answer. Sibling species, species which are morphologically identical but reproductively isolated. And parapatric species are species occurring in separate areas. They share a common boundary. Okay, so that is the answer. A2, B1, C4 and D3. Then next one. Arrange the events in the correct order of sequence. Cleavage, cortical rotation, mid-blastular transition, gastrulation. So correct sequence you have to find out. So first will be cortical rotation. So B first. So it is the rearrangement of the egg cytoplasm before cleavage, establishing the future dorsal ventral axis. That is cortical rotation, and it takes place before cleavage. So first will be cortical rotation, then comes cleavage, and then mid-blastular transition, that is the stage before gastrulation. So C and D. So the answer here is option 3, B, A, C, D. Then next one. Okay. Arrange the following in the correct sequence of DNA packaging. So DNA packaging, the simplest level is the double-stranded helical DNA. From there you have to go to the DNA complexed with histones, that is nucleosome, and each nucleosome consists of 8 histone proteins around which DNA wraps 1.65 times. Then comes the chromatosome, that is the nucleosome with H1 histone. Then the nucleosome fold up to produce the 30 nanometer fiber. Then again forms loops and result in 300 nanometer length. Again compressed to 250 nanometer fibers, and there will be, in the end, there will be tight coiling to produce the chromatid of the chromosome. This is the sequence of DNA packaging. Now see here. So first will be histone dimerization. Okay, that will be the first step in DNA packaging. One minute. Okay. Because histones are the building blocks for nucleosome. So after D, it has to be nucleosome formation B, but they have given C here and then B. As per this diagram also, It is first nucleosome, then chromatosome. But here they have given DCBA. It might be a printing mistake. Okay. Actually, it has to be DBC and then that DNA scaffold. So, since they have given this as the answer and it doesn't have DBCA as the option, I am going with the option 2. DCBA. Then next one, arrange the following animals on the basis of their phyla according to increasing level of complexity. So here the first one will be Euplectula, that is D, which is a porifera, then Physalis, which is Nidaria, E, then comes Nereis, then comes Dentalium, and finally Scolopendra. So D, E, B, C, A, that is option 1 is the answer. Then next one, arrange the following evolutionary events in the history of horse evolution chronologically. So first one was Hyacotherinium, C. Then comes Orohippus, then comes Myohippus, and then finally Merychippus. So CBAD. CBAD, that makes 4 as the right answer. Then which of the following statements are true? Differential gene expression can occur at the level of transcription. Need not be always. It can occur at post-transcriptional regulation, translational regulation, post-translation also it can occur. So A is not correct. RNA polymerase binds to the promoter region to initiate transcription. Yes. B is correct. Enhancers regulate transcription. Yes. Enhancers are can increase transcription. Okay. So C is correct. Transcription factors can recognize specific DNA sequence on DNA heterochromatic region, not on the heterochromatic region. Heterochromatic region is compact structure and transcription factors cannot access DNA sequence in heterochromatic region, whereas they can access DNA sequence in the euchromatic region, not in heterochromatic region. So that makes D also as wrong answer. So B and C is the answer. Answer is option 2. Then which two embryological structures together form together form the bilaminar disc? Epiblast and hypoblast is the answer. What is cryptotrophoblast? It is a type of cell in the placenta that help the embryo to attach to the uterine wall. Okay. Then what is syncotrophoblast? It is a layer of multinucleated cells that form the outermost part of the placenta, and it is involved in the transport of nutrients, gas and waste between the mother and the fetus. Okay, so that's it. Then coming to the next question. Cytoskeleton of eukaryotic cell consists of. So cytoskeleton in eukaryotic cell, it is having microtubules, microfilaments and intermediate filaments. Here only microtubules and microfilaments are given. That is B and D is the answer. So answer is option 1, B and D only. Then next one. Which of the statements are correct with regards to infrared spectroscopy? So functional groups can be identified by looking in the fingerprint region of the spectrum. Yes, infrared spectroscopy allows you to identify functional groups that are present in a compound. But the statement is the functional groups peak are observed in the functional group region which is adjacent to the fingerprint region. So by looking in the fingerprint region, you are not going to get any functional group. So that makes first option incorrect. Then next one. It shines infrared light on a compound and records the position. No, the spectrum cannot be used to find the position of a group. So 2 is also not correct. It is useful in determining the size and shape of compounds' carbon skeleton. It cannot provide information regarding the compound's carbon skeleton. So 3 is also not correct. When the infrared light frequency matches the frequency of bond vibration in a molecule, a peak is recorded on the spectrum. That is correct. So 4 is the right answer. Then match list 1 with list 2, phase and characteristic of the phase. Lag phase. So adoption to the nutrient environment. A3. Oh fine. A3 is there only in option 4. Then a log phase. Binary fission at the maximum rate. Stationary phase. Number of new cells equal to dying cells. And decline phase. Dying cells are higher than the dividing cells. So answer is option 4, A3, B4, C2, D1. Then a technique with the connecting or purpose. DNA footprinting, it is used for finding out DNA binding proteins. That is like your RNA polymerase, it binds to the promoter. Promoter is a part of the DNA sequence. So DNA binding protein. RNA polymerase is a protein. So to which promoter region this RNA polymerase is binding, you can find out by DNA footprinting. So A3 is there in 2 and 3. Okay. Yeast 2 hybrid system, protein-protein interaction, B1. Okay, B1 is there only in option 2. Then DNA fingerprinting, that is VNTR, variable number of tandem repeats. Then SAGE is transcriptome analysis, D4. So that makes option 2 as the answer. A3, B1, C2, D4. Then next one, structure with the function. Ascending loop of Henle, it is for sodium is actively thrown out. A2, okay, they have tried to confuse you by giving in 1, 2 and 3. Okay, see, next one. Descending loop of Henle is passive entry of sodium. So B4 is there in 1 and 2 but not in 3. So 3 is not the right answer. So we have to see between 1 and 2. Proximal tubule, it is for reabsorption of glucose, C1. And collecting duct is diffusion of water, D3. So that makes option 1 as the answer. Then next one. Sequentially arrange the following steps involved in blood clot formation. So first one will be reduction of blood loss by initiating a vascular system. That is correct. So E is the first one. Okay. So E is the first one in 1 and 3 options. Okay. Next one will be adhesion and aggregation of platelets on damaged vessels, which they have not given in 1 and 3 options. They have given D in one case and B in the other case, that is conversion of prothrombin to thrombin, which is not going to happen as soon as there is reduction of blood loss by initiation of vascular system. So 3 cannot be the answer. So you have to forcefully go for option 1. I don't know whether how far it is correct, because as far as I have seen and read, it is adhesion and aggregation of platelets on damaged vessels. That has to come after E. But here it might be a printing mistake also. Okay. Then after that comes, as per me, it is prothrombinase formed by extrinsic or intrinsic pathway. Then conversion of prothrombin to thrombin and conversion of fibrinogen to fibrin. So since in your answer key 1 is given as the answer, I don't have any other option. So as per me it is E, C, D, B, A. Then next one. Estrus and menstrual cycles are two types of cycles that occur in female mammals. Okay. Menstrual cycle occur in primates and humans, and estrus cycle in non-primates. Both cycles begin after sexual maturity in females. Reproductive hormones induce these cycles in females. This cycle happens in order to prepare the female mammal for holding a child during pregnancy. The primary difference between estrus and menstrual cycle is, okay, the endometrium shed by the uterus during the estrus cycle is reabsorbed, but the shed endometrium is excreted from the body during menstrual cycle. Yes, that is the primary difference between estrus and menstrual cycles. So 1 is the answer. Behavioral changes during estrus cycle are much less pronounced effect on estrus cycle than they do on the menstrual cycle. Behavioral changes during estrus cycle are much less pronounced effect on estrus cycle than they do on menstrual cycle. Okay, so that is also not correct. Then season and climate have less pronounced effect on estrus cycle than they do on menstrual cycle. Seasonal and climate, they can affect both estrus and menstrual cycles. Then most estrus cycles are of much longer duration compared to menstrual cycles. Actually, estrus cycle vary greatly across species. You cannot generalize and say that they are longer than menstrual cycle because in some animals it is shorter duration. Okay, so 4 is also not correct. Then which of the following statement about cortisol are correct? It decreases the circulating lymphocyte. Yes, that is correct. It increases during the circulating eosinophils. That is not correct. It decreases the production of prostaglandins. Yes, it has, cortisol has anti-inflammatory effect, and this is achieved by decreasing the production of prostaglandins. Then it inhibits the production of fibroblast. Yes, it alters the immune system response and suppresses the digestive and reproductive system. That is also correct. So A, C, D and E, that makes option 3 as the right answer. Then again, which of the following statements with regard to plasmid incompatibility are true? Now what is this plasmid incompatibility? It refers to the condition where two or more plasmids cannot coexist in a single bacterial cell. Now, when does this plasmid incompatibility occur? That is, when both the plasmids have the same origin of replication, have similar replication control mechanisms, and they utilize the same replication resources. Such plasmids are incompatible. Okay. Now read the options. Plasmids having the same origin of replication belong to the same incompatibility group. That is correct. Plasmids having different origin of replication belong to different incompatibility group, not the same. So B is not correct. Plasmids of the same incompatibility group are compatible. They are not compatible. They are incompatible. They cannot coexist together. Okay, in the same cell. So they are incompatible. So D. So C is not correct. D is correct. So that makes option A and D only. That is option 2 as the answer. Then which organism species have greater diversity in Western Ghats than Eastern Ghats? That is your amphibians. Which of the following is a feature of stable community? Susceptible to invasion by alien species? No. Susceptible to occasional? No. They are not susceptible to anything. They are resistant to occasional disturbances. Yes, that is correct. Then the transition zone in biosphere, it is the outermost zone of the biosphere reserve. 4 is correct. Then next one, migratory species pose special preservation challenge because they are endemic, they are especially susceptible to habitat destruction. No. Their conservation may require international cooperation. Yes, they are migrating from one country to another. When they require habitats in different countries. So that is correct. That is more correct. They are often prone population number decline during their long migratory journeys. No. They reside in biodiversity hotspots that are most susceptible to habitat degradation. No.